In this lecture, we're going to discuss the chromosomal basis of inheritance. Before the lecture begins, make sure that you've read the assigned chapters in the textbook. During the lecture, make sure that you take plenty of notes, rewording the information in your own words. During the lecture, make sure you've minimized distractions by turning off any music or TV or movies going on in the background. Listen in a quiet space and turn off any extra devices. So today we're going to look at the chromosomal basis of inheritance. We're going to look at Mendelian inheritance and its physical basis in chromosomes, and we're also going to look at sex-linked genes. First, let's discuss the chromosomal theory of inheritance. Today we know that Mendel's factors are segments of DNA located on a long chromosome. We can see the location of a particular gene by tagging chromosomes with a fluorescent dye that highlights this gene. For example, in figure 12.1 on the slide, you can see the tubular regions that mark a specific gene on a human chromosome. In this case, this is human chromosome 6. So this chromosome has been duplicated so that the alleles on the chromosome are present as two copies, one sister chromatid. And again, when we're talking about these chromosomes, we're talking about DNA that's been wrapped up. DNA in its simplest form is a double-stranded helical structure. It wraps around the histone core of 8 molecules in total so that this can further condense. Eventually, these wrapped up forms then form loops and folds that compress into a tight coil. This forms the chromosome. Keep in mind, when Mendel looked at these hereditary factors, these were purely an abstract concept when he proposed the existence of these factors in 1860. At that time, no cellular structures had been identified that could house these units, and most biologists were skeptical about Mendel's proposed laws of inheritance. The chromosomal theory of inheritance states, Mendelian genes have specified loci or positions on a chromosome, and that chromosomes undergo segregation and independent assortment. In 1902, Walter Sutton and Theodore Boveri developed the chromosomal theory of inheritance. According to this theory, Mendelian genes have specific loci among chromosomes, and it is the chromosomes that undergo segregation and independent assortment. Now the law of segregation states that the two alleles for each gene separate, and also the law of independent assortment states that the alleles of a gene on non-homologous chromosomes assort independently. Now we're going to look at the genes that are found on our sex chromosomes. Humans and other mammals have two types of sex chromosomes, a larger X chromosome and a smaller Y chromosome. Only the ends of the Y chromosome have regions that are homologous with the corresponding region of the X chromosome. The SRY gene on the Y chromosome is required for the development of the testes. So the SRY strands are the sex-determining region of the Y chromosome. By this system, females are XX and males are XY. Each of them contain an X chromosome, while sperm may contain either an X or a Y chromosome. Other animals have different methods of determining sex. Humans have a total of 46 chromosomes, 44 autosomes, and 2 sex chromosomes. In this image, the male parent has 44 chromosomes and the XY sex chromosomes, whereas the female has 44 chromosomes and 2 sex chromosomes, which are X chromosomes. The male sperm will carry half, or 22 autosomes, and either an X or a Y sex chromosome. Additionally, the egg will carry 22 autosomes and a single X chromosome. Once fertilized, the offspring or the zygote that is produced will have 44 autosomes and either an XX or an XY set of sex chromosomes. Researchers have sequenced the human Y chromosome and have identified 78 genes that code for about 25 proteins. About half of these genes are expressed only in the testes, and some are required for normal testicular function and the production of normal sperm. A gene located on either sex chromosome is called a sex-linked gene. Those located on the Y chromosome are called Y genes. The Y chromosome is passed among virtually intact from father to all sons. Because there are so very few Y-linked genes, there are very few disorders that are transferred from father to son on the Y chromosome. The X chromosome contains approximately 1,100 genes, which are called X-linked genes. The fact that males and females inherit a different number of X chromosomes leads to a pattern of inheritance that's different from the production by genes located on autosomes. X-linked genes follow a very specific pattern of inheritance. For example, if there is a recessive X-linked trait, in order for this to be expressed, a female needs 2 copies of the allele. She must be homozygous. And a male only needs 1 copy of this recessive allele. He will be hemizygous. Because males only have 1 locus, the term homozygous and heterozygous lack meaning when describing their X-linked genes. In turn, hemizygous is used to define the male single copy of the X-linked genes. For this reason, far more males and females have X-linked disorders. However, even though it's characteristic of a female inheriting a double dose of a mutant allele, it's much less than the probability of a male inheriting a single dose. Still, there are females with X-linked disorders. Here are a few examples of disorders caused by recessive alleles on the X chromosome. Color blindness, which is mostly X-linked, Duchenne muscular dystrophy, and hemophilia. Color blindness is almost always inherited as an X-linked trait. So we can use this pedigree to look at and consider how color blindness is an X-linked trait. A colorblind daughter may be born to a colorblind father whose mate is a carrier, because the X-linked allele for colorblindness is relatively rare. However, the probability that such a man and female will mate is very low. In the figure here on the screen, we're looking at the transmission of an X-linked recessive trait, so specifically the red-green color blindness. This is being used as an example in this cross, so the superscript N represents the dominant allele for normal color vision, carried on the X chromosome, whereas the small n represents the recessive allele, which has a mutation in the DNA resulting in red-green color blindness. In females, 40 to 60% of cells express one of the X chromosomes, and in the other cells, the other X chromosome is expressed. The determination of which X chromosome is expressed in which cell is regulated by a gene called the Xist gene. The inactive X condenses into what is known as a bar body. If a female is heterozygote for a particular gene located on the X chromosome, she will be a mosaic for that character. A common example used for looking at X inactivation are calico cats. Now let's review.
The secrets of the X chromosome. These women are identical twins. They have the same nose, the same hair color, the same eye color. But this one is colorblind for green light, and this one isn't. How is that possible? The answer lies in their genes. For humans, the genetic information that determines our physical traits is stored in 23 pairs of chromosomes in the nucleus of every cell. These chromosomes are made up of proteins and long coiled strands of DNA. Segments of DNA, called genes, tell the cell to build specific proteins, which control its identity and function. For every chromosome pair, one comes from each biological parent. In 22 of these pairs, the chromosomes contain the same set of genes, but may have different versions of those genes. The differences arrive from mutations, which are changes to the genetic sequence that may have occurred many generations ago. Some of those changes have no effect, some cause diseases, and some lead to advantageous adaptations. The result of having two versions of each gene is that you display a combination of your biological parent's traits. But the 23rd pair is unique, and that's the secret behind the one colorblind twin. This pair, called the X and Y chromosomes, influences your biological sex. Most women have two X chromosomes, while most men have one X and one Y. The Y chromosome contains genes for male development and fertility. The X chromosome, on the other hand, contains important genes for things other than sex determination or reproduction, like nervous system development, skeletal muscle function, and the receptors in the eyes that detect green light. Biological males with an XY chromosome pair only get one copy of all these X chromosome genes. So the human body has evolved to function without duplicates. But that creates a problem for people with two X chromosomes. If both X chromosomes produced proteins, as is normal in other chromosomes, development of the embryo would be completely impaired. The solution is X inactivation. This happens early in development when an embryo with two X chromosomes is just a ball of cells. Each cell inactivates one X chromosome. There's a certain degree of randomness to this process. One cell may inactivate the X chromosome from one parent, and another the chromosome from the other parent. The inactive X shrivels into a clump called a bar body and goes silent. Almost none of its genes order proteins to be made. When these early cells divide, each passes on its X inactivation. So some clusters of cells express the maternal X chromosome, while others express the paternal X. If these chromosomes carry different traits, those differences will show up in the cells. This is why calico cats have patches. One X had a gene for orange fur, and the other had a gene for black fur. The pattern of the coat reveals which one stayed active where. Now we can explain our colorblind twin. Both sisters inherited one mutant copy of the green receptor gene and one normally functioning copy. The embryo split into twins before X inactivation, so each twin ended up with a different inactivation pattern. In one, the X chromosome with the normal gene was turned off in the cells that eventually became eyes. Without those genetic instructions, she now can't sense green light and is colorblind. Disorders that are associated with mutations of X chromosome genes, like colorblindness or hemophilia, are often less severe in individuals with two X chromosomes. That's because in someone with one normal and one mutant copy of the gene, only some of their cells would be affected by the mutation. The severity of the disorder depends on which X got turned off and where those cells were. On the other hand, all the cells in someone with only one X chromosome can only express the mutant copy of the gene, if that's what they inherited. There are still unresolved questions about X inactivation, like how some genes on the X chromosome escape inactivation and why inactivation isn't always random. What we do know is that this mechanism is one of the many ways that genes alone don't tell our whole story.
So today we looked at the chromosomal basis of inheritance. We looked at Mendelian inheritance and its physical basis in chromosomes, as well as sex-linked genes. If you have any questions about this lecture or the additional information in your textbook, please reach out to your professor through email or office hours or by bringing your questions to class. Additionally, you can visit the class learning assistants who are eager to support your learning in Bio 110. Finally, as you finish up this lecture, combine your notes from the lecture with any notes that you took from your assigned reading. Use the assisted reading questions to prepare you for the quiz and the test, as well as questions at the end of the chapter and at the end of each chapter section.